In Unit 3, we'll be taking a look at the molecular basis of inheritance and gene expression. When we talk about the molecular basis of inheritance and gene expression, we really are talking about the nucleic acids, DNA and RNA, and protein synthesis with respect to gene expression. The information for this unit will be coming primarily from Chapter 13, which looks at the historical work that was done to help us understand the molecular structure of the DNA molecule and the structure of DNA and RNA, and then Chapter 14 that primarily will be dealing with protein synthesis. Before we go too far down the path of talking about DNA and protein synthesis, I want to visit just a little bit about how we came to understand the working portion of the chromosome or DNA. In the late 1800s, there was work being done by an Austrian monk named Gregor Mendel, who was studying pea plants, and his work really led us to the beginning of the process of looking at what controlled for these physical traits or characteristics that we see expressed in living organisms. And so based on his work in the early 1900s, scientists started to take a little bit closer look at essentially the chromosomes. And we knew that chromosomes existed both in prokaryotes as a one-ring chromosome in the nucleoid and as multiple linear chromosomes in the nucleus of eukaryotic cells. And there was also an understanding that these chromosomes were primarily made up of two different types of molecules, those molecules being DNA and proteins. But we didn't know at that point whether it was DNA or protein that was actually the molecular basis for inheritance, where the information was being stored that was being passed on from generation to generation. And so as you look at this diagram, you can see the two components basically that make up the chromosome, those being the proteins, the histones, which help to organize and package the DNA, and then the DNA molecule itself, which we know today is where the genetic information is stored. There was important work being done in the 1930s and the 1940s and up into the 1950s in trying to decipher whether or not proteins or DNA was the molecular basis for inheritance. There was a pretty elegant experiment done in 1953 by Alfred Hershey and Martha Chase that essentially used radioactive isotopes to help to establish that it was DNA and not protein that was where the information was actually being stored in the chromosome. Essentially what Hershey and Chase did was they used a bacteriophage, T2, which is a virus that infects bacteria, to try to determine whether or not it was a protein or the DNA that was carrying the genetic information. And what they did was they radioactively labeled a sulfur element in the protein in this bacteriophage, and they also radioactively labeled the phosphorus, which is in the DNA in the bacteriophage, in the virus. And what they did was trace basically these two elements as the bacteriophage went through the process of infecting the bacterium. Once they separated out the components from the bacteria, they found that essentially the protein that contained the sulfur never actually entered the bacteria, but the phosphorus that was radioactively labeled in the DNA molecule had entered the bacterium. And this provided really powerful evidence that it was DNA and not protein that was acting as the molecular basis for inheritance. It was a component, the part of the chromosome that was actually physically storing the genetic information. Their work led to some important work in the 1950s by several other prominent scientists that you may have heard of. Along with Hershey and Chase's experiment, there was another scientist by the name of Erwin Chargaff that provided some very fundamental important information that helped us to understand the molecular structure of the DNA molecule. Chargaff's work again was being done kind of in the 1930s and 1940s, and essentially what Chargaff brought to the table was that understanding that there was a difference in DNA between species, and this difference in DNA could be related to some of the physical traits or characteristics that we see expressed. Maybe even more important than understanding that there were differences between DNA and different species was Chargaff's understanding that there was a relationship between the percentage of nitrogenous bases in the DNA molecule. And what Chargaff came to understand was that there was an equal percentage of adenine and thymine and an equal percentage of guanine and cytosine in the DNA molecules. These two pieces of information combined are called Chargaff's rules. So by the 1950s, there had been some important work done by several people that helped us to begin to understand the basic characteristics and structure of the DNA molecule. There are several people on this slide, and some of them you may have heard of, that get a tremendous amount of credit, and rightfully so, for basically unraveling the mystery of the molecular structure of the DNA molecule. Two that you may have heard of or are kind of more well known are James Watson and Francis Crick. And Watson and Crick published a paper in 1953 that basically outlined the molecular structure of the DNA molecule. And here you can see them pointing out their model, which we now know as this famous double helix, which is the basis for carrying the molecular information for inheritance. But two other people that played a prominent role in Watson and Crick's understanding of the structure of DNA were Rosalind Franklin and Maurice Wilkins. You can see the X-ray crystallography image that was generated by Franklin while working with Wilkins that Watson and Crick both had access to, that eventually led to, I think, helping them build this three-dimensional model of the molecular structure of DNA. And so there was a lot of work that was done before Watson and Crick, but certainly Watson and Crick deserve a lot of credit for putting the pieces together. And in the early 1960s, Watson and Crick and Wilkins all won the Nobel Prize for describing the molecular structure of DNA. Unfortunately, by that time, Rosalind Franklin had passed away and was not eligible for the Nobel Prize. So if we fast forward today, and over the last 20, 30, or even 40 years, we know that research in DNA and molecular research at the cellular level has just been moving incredibly fast. And so by the 1980s and 1990s, we had started to develop the ability or the technology to actually manipulate DNA, and had started to do gene splicing or gene bashing, where we could take sequences of DNA from one organism and splice that sequence of DNA into another organism and see that gene expressed. And so, for example, here you can see a firefly gene being expressed in a tobacco plant and a jellyfish gene being expressed in a pig. And you might say, well, what kind of practical applications does that have? Well, a lot of the GMOs today, the genetically modified organisms that we see today, have been done to manipulated to help us provide food or medicine, and have really changed not just science and biology, but our society overall. And so a lot of the really interesting research today in terms of like gene splicing and sequencing genomes and stem cell research and cloning is based on the foundation and the work that was done in the 1940s and the 1950s by some of these early pioneers that were working on DNA. The GMOs get a lot of kind of press now, and genetically modified organisms, especially plant-based GMOs, have been around with us for a long time. And so things like BT corn, which has basically a gene spliced into it from a bacterium that gives it almost like an internal resistance to insects or internal pesticide, has been used for some time now. And one of the things that we're seeing more recently is GMOs, or genetically modified organisms, in the animal side of our food supply. And one of the first animals that they're looking at introducing as a GMO, or genetically modified organism, is an Atlantic salmon. And this is you know, I think very close to becoming the first animal-based food source that has been genetically modified that will be introduced into our food supply. There's a lot of questions, and in general the research I think has not shown that there's necessarily a lot of health concerns directly related to GMOs in terms of consumption, but there are some real concerns in terms of the ecology of how these GMOs might affect different systems, different ecosystems. And it's still early, and I think there still needs to be, you know, research done and continue to be done to fully understand the impacts, all of the impacts of these GMOs. And our ability to manipulate basically DNA, really the ingredients of life, comes with it, you know, I think some incredible potential in terms of dealing with, you know, supplying food and new medicines, and really helping to make our society a better place. But along with it will also come some important ethical and moral questions that we will also have to address as we kind of move into this brave new world. So having set the kind of the historical stage, and maybe giving you just a little bit of background about where we are today, I want to take a more traditional approach to looking at the basic structure and function of nucleic acids, and then eventually look at protein synthesis in a little bit more detail. So as we mentioned earlier, the nucleic acids basically are DNA and RNA. And DNA stands for deoxyribonucleic acid, and RNA is ribonucleic acid. The deoxy and the ribose basically, or deoxyribose and ribose, both refer to the sugars that we find in these nucleic acids. The nucleus, kind of nucleic, refers to the fact that it's found in the nucleus of, or near the nucleus of, eukaryotic cells. And these are acids, but don't think of them as like a strong acid, but they do have a tendency to be proton donors. So DNA is a chemical information system for the cell, directing the cell's activities, and we'd mentioned this before. It really controls three things. It's the basis of inheritance. It's how we transfer or pass on genetic information. Controls cell division, that being mitosis and meiosis and sexually reproducing organisms. And then the third major process that DNA is responsible for controlling is protein synthesis. And those two forms of nucleic acids again are DNA and RNA. So if we go back to the two basic types of cells that we've discussed before, we know there are prokaryotes and eukaryotes. DNA in prokaryotic cells is found as this one-ring chromosome just loose in the cytoplasm called a nucleoid. And you can see to the left on this slide basically a prokaryote, a bacteria that's undergone lysis, and all of that kind of white stringy looking substance surrounding the cell is actually the DNA. It's incredible how efficient and how accurately and how much information can actually be stored on these nucleic acids. It just kind of boggles the mind. And in terms of like our own genome, and remember prokaryotes have a much smaller genome, maybe up to 5,000 genes or so, but in our own genome, you know, we have approximately 30,000 genes that are made up of like 3 billion base pairs. And when you think about the size of you know, a eukaryotic cell, and you know, let alone a prokaryotic cell, that is just a tremendous amount of information, and it's very efficient in terms of storing that information. So again, based on the work of Hershey and Chase, we know now that it's DNA that constitutes the working portion of the chromosome, and the proteins, the histones associated with the chromosomes, really are just there to help with the basically the packaging and maintaining the structure of the chromosome. The other type of nucleic acid, ribonucleic acid or RNA, there are 3 types, and the 3 types of RNA we'll get into in a little bit more detail later, but those are essentially messenger RNA, transfer RNA, and ribosomal RNA. The other cell that you see to your left there, you have the prokaryotic cell above and then the eukaryotic cell just below it. So the DNA in eukaryotic cells, we know, is inside of that membrane in the cell, the nuclear membrane, and inside of that nuclear membrane there are multiple linear chromosomes. And eukaryotic cells just have a little bit larger library than prokaryotic cells. They can store more information, which gives them the ability to make more proteins. The RNA, and those 3 types of RNA that we talked about, really depending on the type of RNA and what's going on with respect to the cellular processes, might be found in the nucleus, in the cytoplasm, or at a ribosome, interacting with DNA to kind of help drive these cell activities. This gives you a closer look at the structure of the DNA molecule, and we know that DNA is a double-stranded molecule, and a lot of that information again was worked out in the mid-1900s by Watson and Crick and others. But the molecule itself is 2 strands, and the outer portion of the strands are made up of basically repetitive sequences of phosphate and sugar groups, and then connecting those 2 strands are the nitrogenous bases, and it's the nitrogen bases where the information in the DNA molecule is actually stored. Those nitrogenous bases are held together by hydrogen bonds. And the nitrogenous bases, the 4 thymine, adenine, cytosine, and guanine, have a definitive way of pairing, and there's a specific number of hydrogen bonds between those nitrogenous bases. If you take a closer look at the double-stranded molecule in the middle of the diagram, you can see that adenine and thymine always combine together, and there are 2 hydrogen bonds between adenine and thymine. And cytosine and guanine always combine together, and there are 3 hydrogen bonds between those 2 nitrogenous bases. It's the unequal number of hydrogen bonds between the nitrogenous bases and the kind of unequal pull between these hydrogen bonds that cause the DNA molecule to twist or coil into that all too familiar term now that we know of in terms of the DNA structure as being a helix or a double helix. And it's a pull of those hydrogen bonds that causes those two strands to twist or coil into that helix. Basically, in the nucleus of eukaryotic cells, one continuous strand of DNA, and again those associated proteins, the histones, make up a chromosome. In your normal body cells, your somatic cells, you have 46 chromosomes that are occurring in 23 pairs. There are some similarities between DNA and RNA, and one of those similarities is really the structural unit. Both DNA and RNA are considered to be polymers, and polymers basically are large molecules that are made up of many smaller repetitive sequences of molecules. And so the structural unit of both the DNA and the RNA molecules, the nucleic acids, is called a nucleotide. And a nucleotide is composed of three things. Basically, the three things that make up a nucleotide are a phosphate group, a sugar, and then a nitrogenous base. And that's true for both DNA and RNA. So we take a closer look at these nucleic acids, we'll see that there are some differences, both molecular and structural differences between DNA and RNA. But one of the things that is, I guess, similar between the two molecules is that, the basic repetitive sequence of molecules of these phosphates, sugars, and nitrogenous bases, these nucleotide sequences, are responsible for making up these larger polymers or molecules of both DNA and RNA. Let's take a little bit closer look at the nitrogenous bases in these nucleic acids. There's two major groups of nitrogenous bases, those two groups being purines and pyrimidines. And if you look to the left, you can see that the purines, adenine and guanine, are a little bit larger molecules. They have an extra carbon ring associated with them. And I just remember that around here, one of the things that we do is farming or agriculture. And I remember that ag is really big around here. And so I remember that the larger molecules are adenine and guanine. And then the second group of nitrogen bases are called pyrimidines. They're a little bit smaller molecules. They don't have that extra carbon ring associated with them. And the pyrimidines are thymine and cytosine if we're talking about DNA. But in RNA, uracil will replace thymine. So there's actually can be three different pyrimidines spread across the two types of nucleic acids, DNA and RNA. Remembering that uracil replaces thymine when we're talking about RNA as opposed to DNA becomes important when we take a closer look at protein synthesis and how this information is read and eventually converted into a protein. Now, having said that, these nitrogen bases or nitrogenous bases have a specific way of combining. Purines always combine with pyrimidines. And if you look, you can see that adenine will always combine with thymine, and there is a way to kind of ensure that you have the right complementary nitrogenous base, and that is based on the number of hydrogen bonds between the two bases. And so adenine and thymine only fit together correctly with 2 hydrogen bonds. And you can see that guanine and cytosine are the other pair of complementary nitrogenous bases, and they only fit together with 3 hydrogen bonds. And so that helps to ensure that when you're copying DNA or when you are moving information from DNA to RNA, that you get the right complementary base as this information is kind of being copied and handled and moved by the cell. So remember that purines always combine with pyrimidines, adenine with thymine, guanine with cytosine, and if we would happen to be talking about RNA, then thymine would be replaced with uracil. And it's that unequal pull of those hydrogen bonds that causes that twisting or coiling structure called the helix in the DNA molecule. So both DNA and RNA structure are composed of these kind of repetitive sequences of basically phosphates, sugars, and nitrogenous bases. Remember, it's a phosphate, a sugar, and a nitrogen base that make up a nucleotide, but the phosphate and the sugar really are just structural components of the DNA molecule. They aren't storing any information. They're just providing the backbone or the structure or the support for the molecule. It's the nitrogen bases, these sequences of adenine and thymine and cytosine and guanine, and the order that these bases are found in the nucleic acids that dictate what the proteins will look like. You know, what sequence of amino acids will be put together in what order. And so it's the nitrogen bases that are the key in terms of storing information in nucleic acids. If we do a comparison and contrast between the nucleic acids, DNA and RNA, DNA is a double-stranded molecule. It has a sugar called deoxyribose, and when you see that D, that means without, and then the oxy is oxygen. Now that does not mean that deoxyribose does not have oxygen in it. That sugar does have oxygen in it, but it has one less oxygen atom in it than the sugar that we find in RNA and the ribose. But that, again, that's one of the differences between DNA and RNA, is a difference in those sugars, and then also a difference in the nitrogenous bases, where DNA uses thymine as one of its nitrogenous bases. RNA is single-stranded, and it uses ribose as its sugar. And remember that in RNA, uracil will replace thymine as one of those nitrogenous bases. Don't forget the two major groups, purines, adenine and guanine, and pyrimidines, cytosine and thymine in DNA, and then uracil has to get thrown in there if we're talking about RNA as a replacement for thymine. This just kind of shows you know, a double-stranded nature of the DNA molecule as opposed to the single-stranded RNA structure. And you might also notice that you won't see any thymine in the RNA molecule. You'll see uracil where you would see thymine in DNA. We're going to talk a bit about DNA replication, and that will lead us eventually to a little bit more specific conversations about protein synthesis. And one of the things that happens from time to time is people will kind of mix these processes up, and you want to keep these separate. These are different things. I think understanding DNA structure and DNA replication is important to building a bridge and understanding protein synthesis, but remember that these are two different processes. And one of the first things that we need to look at is, you know, when and why does DNA replicate? Up above you can see what's called a karyotype, and this is a karyotype of a somatic cell, a human somatic cell, and so you can see the 46 chromosomes there that are occurring in 23 pairs. One of them is a normal male karyotype, and if you look at the 23rd pair there, you can see those are the gender pair of the basically the genome, and this particular individual has an XY for that pair, so that means that that's a male. A female would be XX. The far right one is actually a karyotype of an individual that they call it trisomy 21, or a more common term is Down syndrome, and once in a while you can have basically issues with chromosome counts if you get an unequal division of the chromosomes in the gametes in the sperm or the egg, and that can create issues, developmental issues, and some of those things. But let's go back to when and why DNA replicates. DNA would replicate right before cell division. And so remember, two types of cell division, mitosis and meiosis. And in humans, in our normal body cells, our somatic cells, and even in the beginning cells in meiosis, we have 46 chromosomes. And so before a cell undergoes cell division, it will actually replicate or double each one of those 46 chromosomes. So for a short period of time before the cell divides, you'll have 92 chromosomes in the cell. In mitosis, you just go through one division of the nucleus and you end up with basically 46 in the two daughter cells. In meiosis, it's a little bit different. You actually reduce the chromosome count by 1/2 in the gametes by going through two divisions of the genetic material. And we'll hit on that a little bit later when we look at cell division and mitosis and meiosis. But when DNA replicates basically is right before cell division. And then why? It's just one reason is normal growth and development. So we all start out as a single cell, the zygote, after the fusion of the gametes, the sperm and the egg. And so to go from that 1 cell to 2 cells to 4 cells to 8 cells to this incredible organism that's made up of trillions of specialized cells, we have to go through this process of DNA replication and cell division. Your cells don't last forever, and so we have to replace damaged and worn out cells. And this is kind of an ongoing process. Some organisms will replicate DNA for a type of reproduction and asexual reproduction in some organisms, prokaryotic cells, bacteria, protozoans. Some of these organisms that do asexual reproduction obviously would need to go through basically DNA replication. Now, the karyotypes above don't necessarily reflect a mutation in the copying sequence. It's more of an issue in terms of the number of chromosomes, but mutations can and do occur when we make mistakes in the copying of the DNA. And so we can have things like substitutions or deletions, or even something that's called a frame shift, where you might get off one base pair as you're copying the information, and those mutations really can have 3 different outcomes. If it's not in a critical area of the DNA in terms of the coding for an amino acid, or which eventually is going to dictate the structure or the function of the protein, it may not have any effect at all. If you have major mutations, generally those types of mutations are detrimental and can be fatal. But once in a while those mutations can be beneficial. It might give an organism a competitive advantage, a little bit thicker coat in a cold climate, the ability to maybe more efficiently process oxygen and become more energy efficient. Those types of mutations tend to give those organisms a competitive advantage. You know that those organisms tend to have a higher survival rate. They reproduce, they pass on those characteristics, and so those types of mutations can and do contribute to natural selection and moving organisms to higher levels of fitness. When you look at DNA replication in theory, it's really pretty straightforward. Essentially, this is a double-stranded molecule. You might notice that there is a 5' and a 3' side to each strand, kind of oriented in opposite directions, and that plays a role when we start to talk about how basically the DNA molecule is copied and what direction it's copied. The molecule itself is what they call anti-parallel, and so on one side you have a strand running from 5' to 3' from the top to the bottom, and then on the opposite side kind of the reverse. That 5' and 3' business basically refers to which sugar the molecule that the phosphate group is attached to on that backbone. And there are some videos available that will kind of point that out a little bit more in terms of what that 5' to 3' orientation means. But basically, when we copy the information, we build the information in the 5' to 3' direction. And you can see here, essentially, when we replicate DNA, the hydrogen bonds between the double-stranded DNA molecules separate, and then we bring in complementary base pairs in the 5' to 3' direction, build 5' to 3' for the daughter strands. If we don't make any mistakes, essentially we end up with 2 exact copies or replicates of the original strand or the parent strand, and 2 duplicate copies of the original double-stranded DNA molecule. This process of DNA replication, remember, is going to happen right before the cell divides, and for those reasons that we had mentioned earlier, one of the first things that has to happen is that the hydrogen bonds between the nitrogenous bases need to separate. And there is an enzyme that does that. That enzyme that separates or breaks those hydrogen bonds between the complementary nitrogenous bases is called helicase. If you ever see that suffix ASE in biology, that's telling you that that is an enzyme. So if you see ASE on the end of a term in biology, that's telling you that's some type of enzyme. Helicase, amylase, polymerase, whatever it might be. If you see ASE, that's an enzyme. That's a protein that's catalyzing or speeding up a chemical reaction. In this case, helicase, the prefix hela is referring to the double helix. And so that's telling you what that enzyme is working on. When we begin the process of DNA replication, the enzyme helicase comes in and breaks the hydrogen bonds between the complementary nitrogenous bases. As the double helix unwinds, new complementary nucleotides basically will move into position for the 2 parent strands. The enzyme that helps to reestablish the hydrogen bonds to these reforming duplicate strands or double-stranded DNA molecule is called DNA polymerase. And again, you can see that ASE on the end of that that term, which indicates to you that that's an enzyme. So in practice, pretty straightforward stuff. We separate basically the double-stranded molecule. We essentially break the hydrogen bonds using the enzyme, the hydrogen bonds between the nitrogenous bases using the enzyme helicase, and then we come in and we build the daughter strand, the complementary strand for the 2 parent strands, bringing in complementary nitrogenous base nucleotide sequences, and the enzyme that reestablishes those hydrogen bonds is DNA polymerase. Now, of course, nothing can ever be quite that simple, and so when we take a closer look at what actually happens during DNA replication, in eukaryotic cells you get what are called replication bubbles, and in eukaryotic cells you get multiple replication bubbles. And so remember, we have 3 billion nitrogenous bases to copy in the human genome. So instead of just starting at one end and breaking the hydrogen bonds and moving along in one direction, for the entire length of the DNA molecule, you get the formation of these replication bubbles. And so DNA polymerase will come in and it will separate the complementary strands of the DNA molecule by breaking the hydrogen bonds between the nitrogenous bases, and then you start the process of building basically both strands kind of going in opposite directions and building 5' to 3'. You have what's called a leading strand, and the leading strand moves forward essentially continuously, just adding complementary nucleotide sequences to the daughter strand as it kind of moves down the parent strand, using it as a template. And these are going in opposite directions. Where the DNA molecule has been pulled apart at the ends, that's called a replication fork. And so the leading strand will just kind of continue down that parent strand as a template, but the lagging strand, the strand that's kind of on the opposite side, is always just a little bit behind. And you have to think that that leading strand is, it's kind of in there, and there's not that much room basically to copy the information, and so the lagging strand will be running in the opposite direction and building kind of in little short pieces or snippets or sequences to allow the leading strand room to kind of move forward as it's basically copying the opposite side in these short little snippets or pieces called Okazaki fragments. And so you have the leading strand basically being built 5' to 3' on both sides of the DNA molecule, moving in opposite directions, and then you have the lagging strand, which is always just a little bit behind, that is being built in these kind of short snippets or small pieces called Okazaki fragments. And this just continues on at multiple locations in eukaryotic cells at these points of replication called replication bubbles, until you completely copy all of the information essentially from the from both parent strands. Prokaryotic cells are just a little bit different. Prokaryotic cells just basically have one replication bubble, and they copy that one ring chromosome, and smaller genome, and so they essentially start at one replication bubble and copy in opposite directions until they have copied the entire nucleoid or one ring chromosome. Eukaryotic cells have multiple points of origins of replication. Once all of the information has been copied, you end up with two duplicate double-stranded molecules, double helix molecules, resulting from the original DNA molecule. And as we mentioned earlier, you have 46 chromosomes in the nucleus of your normal body cells or somatic cells. After DNA is replicated, you've copied all 46 of those chromosomes for a short period of time. You have 92. Now you have enough genetic information for both daughter cells, and so cell division can go ahead and proceed, with each daughter cell getting essentially a complete copy of the genetic information that it needs to be able to function. Here you can see a little closer view of a replication bubble showing the leading strands running in opposite directions, kind of being followed by the lagging strand copying in these short sequences called Okazaki fragments. Remember that the information basically on the daughter strand is being built 5' to 3' as it reads the parent strand 3' to 5'. And this is it can be a little bit difficult to understand. If you have your textbook, you may want to take a closer look at this particular section, or maybe get on a YouTube video and something like Khan Academy and take a little bit closer look at DNA replication. Essentially, this wraps up the information in chapter 13 on nucleic acids, DNA, RNA structure, and DNA replication. And so this is a kind of a good place to take a break for this part of Unit 3, and we'll start up again with the second half of Unit 3, taking a look at protein synthesis, which is coming primarily from chapter 14 in your textbook.